I hope you've all had the opportunity to enjoy lunch there and are ready for the next session. Not too sleepy, get the coffees in. So welcome to session 3. It's on Agri-Biotech. I promised to not over-introduce it because our first speaker has a train to catch, so I'll keep it pretty brief. Agri-Biotech is, in my opinion, sort of one of the most rapidly evolving fields in the industrial biotech landscape. And we've got 3 great speakers here in this session, 2 academics, 1 lightning talk from academia. And I hope you're going to be able to see through what they have to say, how sort of engineering biology is informing innovation in sort of products and processes, and how protein engineering, making peptides, molecular farming is creating new sustainable molecules and polymers that hopefully will feed into industry. So with that, I'd like to hand over to Professor Elaine Fitches from Durham University. Elaine has worked for many years on creating novel fusion proteins, and her work is hopefully aiming to replace insecticides in a much greener way. I'll hand over to you, Elaine. Thanks.
Thanks very much, Natalie. Need to work out if I need to use my glasses or not. Okay, so I'm going to speak. Hope everyone can hear the acoustics are pretty good in this room. Thank you very much for the invitation. I am from Durham, so thank you very much for letting me invade your space. And as Natalie said, I'm going to talk to you a little bit today about how we've done some research we're interested in exploiting naturally occurring peptides to develop novel protein-based bioinsecticides, and we use yeast as our biofactory. Okay, just a very short introduction. Why do we need new pesticides? Pesticide resistance is a real growing problem. We've got hundreds of insect pest species that have now developed resistance to insecticides. Very often this is to classes of insecticides rather than 1 or 2. At the same time, we have the withdrawal of an increasing number of pesticides due to their negative impacts upon beneficials or pollinator species, for instance. So that's 2 things that work together to make the farmer's job really hard. So we do have a pressing need for new insecticides with novel modes of action that have vastly reduced impacts upon the environment and ecosystem biodiversity. The global bioinsecticide market is now growing. Biopesticides are becoming more and more routinely used by farmers. It's taken quite a few years for this to take off, but they've now got much more confidence in using these new products. Okay, so nature, there's a whole world of different proteins and peptides in nature that are insecticidal in their activity. And these can range from very big proteins like Bacillus thuringiensis, BT, that I hope most of you have heard of, which can be up to 150 kilodaltons. BT works against certain insect pests, not all. It doesn't work against sapsuckers, for instance, but it essentially works by punching holes in the gut of the insect following ingestion. And then you can go way down to the plant cyclopedes that are very small. We're particularly interested in small, we call them inhibitor cysteine knot motif peptides. They're very structurally confined and they're very stable to changes in pH and temperature, which makes them very suitable for scale up. And we've done a lot of work with venom-derived toxins. Many of these are ICK peptides. So the pros in using naturally occurring insecticidal proteins and peptides, they can be insect-specific. Venom-derived toxins can be very potent when they're injected, and they're readily degradable in the environment, unlike chemical insecticides residues can remain for quite some time and have toxic impacts on ecosystems. The cons associated with this is toxicity is often chronic. Proteins by nature are slower in their action compared to chemicals, and therefore their efficacy is often too low to achieve viable control in the field. The peptides can be prone to very rapid breakdown following ingestion or topical application by the insect pest. So ultimately, many of these peptides require delivery to their target sites of action to be orally or topically effective. And this is where our platform, we call it a platform delivery system, comes in. So as I said, we're interested in working with very small peptides. Sorry, I'm only looking at half the audience. What we do essentially is we fuse them to what we call a carrier protein. This protein actually is from snowdrop bulbs. It's called Galanthus nivalis agglutinin. It's a lectin. By nature, a lectin, it binds to specific sugars. GNA binds specifically to manosylated proteins. And it turns out that insects have an abundance of these proteins, whereas we don't. So it's a very sticky molecule, if you like. So the concept is very simple. We fuse a toxin to the carrier protein. We make a recombinant fusion protein. When the insect ingests that fusion protein, the carrier binds to proteins that line the insect gut epithelia, transports the toxin across to the circulatory system. So it can access numerous membranes within the circulatory system. In addition to that, we know that GNA also binds to the nerve tract of Lepidopteran larvae, so we also think, for instance, for venom, neurotoxins, it further delivers it to its site of action that way. And what we found is that fusion proteins, orally and by contact, are significantly more effective than the toxins on their own. So it's a fusion technology. So it's a simple concept, and this is patented. How do we produce these fusion proteins? We use yeast, conventional restriction ligation cloning. Obviously, there's a lot of different ways that you can combine a carrier and a toxin, and we now use AI to help us design recombinant fusion proteins with a high confidence of correct folding. When you join two proteins together, there is no guarantee that both components will fold. And both of these components, the carrier and the toxins, must be correctly folded to be functional. So conventional molecular cloning, yeast transformation, and then we grow the positive colonies up in a benchtop fermenter. This clearly allows us to reach really high cell densities. We can control pH and oxygen and so on. We use an in vitrogen system, which means as the cells make the protein, they spit it out of the cells, so we don't have to burst open cells, we just need to spin the culture out. And then we end up with this green culture supernatant. There's not many other proteins there. What I should have said is we also incorporate a tag, so in the lab we can purify the proteins just using nickel affinity, so it's a very simple purification process. We then desalt the eluted fractions and we freeze dry, so we end up with a powder product. Now that product is not all protein. Yeast tends to hyperglycosylate, so there's some sugars associated with that. Turns out this is actually quite good for us because it stabilizes the protein. We can keep these proteins in the fridge for 4 or 5 years and they still work. We then go on clearly to do some characterization. So this is SDS page, so we can see how pure our proteins are. We do Western analysis, so we've got anti-GNA antibodies, anti-His antibodies, anti-toxin antibodies in some cases. So we're pretty confident that we have what we want. We then do LC-MS analysis, and that gives us the sequence of our protein products. So we know exactly if the sequence is right. Again, if you're working with these ICK peptides, if you get one amino acid wrong, you can end up with a non-functional protein. So it's really important to understand what you're working with. Okay, so this has gone a little bit awry. Okay, spider venom. Spider venom is a great source of insecticidal peptides. Of course, in nature, spiders prey predominantly upon other insects or other spiders. So the venom contains numerous toxins that target insect nervous systems. And and they can be very selective and potent. And I said many contain these highly stable ICK structural scaffold. We've done quite a lot of work with the funnel web spider hexatoxin family. I should point out, we look at all the really nice discovery science that other scientists do, and we take the very well characterized peptides. And clearly we take the ones that are non-toxic to mammals. Okay, I'm not going to talk too much about venom toxins today. I just want to talk about commercial viability. Vestron, an American company, have proved that this is commercially viable. They commercialized the very first spider venom toxin several years ago. And it's from, if you can see the sequence at the top, HV1H. This has a novel mode of action. It disrupts nicotinic acetylcholinesterase receptors. And they sell it as a contact activity spray application. To target certain Lepidopteran coleopteran pests, they recommend co-application with BT. So this is their delivery system. So they're relying on BT to punch the holes in the gut, and that allows their toxin to go up. So I guess quite cheekily, we took the sequence, we made a fusion protein with GNA, and in case you're interested, there's a couple of papers recently where we've shown that when you fuse this commercialized toxin to GNA, you get significant enhancement in oral and topical efficacy against aphids and psyllids. We've done quite a bit of work with psyllids recently. They are a major vector of a bacterial pathogen that causes citrus green, which is a really big problem for the citrus industry. Okay, so what I want to talk a little bit today about is some work we've done with a pea seed protein. Again, another ICK peptide, known as P-albumin peptide, PA1B. This is the only known proteinaceous inhibitor of membrane-bound V-ATPase rotary proton pumps. Discovered many, many years ago by the late Professor John Gatehouse, who was my PhD supervisor and long-term collaborator, fantastic guy to work with. So he discovered this way back in 1985. And it was patented around that time because it showed some insecticidal activity in its own right against certain insect pests. So it's orally toxic to some insect pests, non-toxic to mammals, clearly we eat a lot of pea seeds, and very stable. So what does it do? So it's rotary pumps were quite complex kind of assemblages of different proteins and are involved in active ion transport across epithelia, whether these be in the gut, the insect gut, the neuronal system, salivary glands, and so on. So it's responsible for organelle acidification, membrane energization, and a whole load of development processes. So it's involved in multiple processes, which to me suggests it's a great candidate for developing as a biopesticide. And what I should say, I don't know if you can see, PA1B binds to one of these subunits in the rotary pump, and it stops the pump turning around. Okay, so as before, we produced recombinant P-albumin peptide and a fusion with GNA. It turns out that in nature, the P-albumin peptide produces two proteins, and in plants, it's cleaved. In yeast, you have to use both PA1B and the adjoining partner, PA1A, to get a functional PA1B peptide. We're not quite sure why. I think it's something to do with helping it fold. So I hope you can see here. So obviously we've got the predicted structures on either side. So we've got STS page. This is gel stain for total proteins. We've got nice clean protein products. We've got immunoblot analysis, so reacts with anti-His, anti-GNA antibodies. And we've got the LCMS data to confirm that we have what we wanted in the first place. Okay, so then now we've gone on and we've looked at oral activity. We do aphid artificial diet feeding assays in the laboratory. Aphids are easy to keep. This is a really nice way of putting known amounts of protein into the diet and then establishing how toxic the proteins are. We work with two different aphid species, pea aphids, specialist, as the name suggests, and generalist, which is peach potato aphid. Why? Because peach potato aphid tend to be more resilient to insecticides, chemical insecticides, and proteins. To do with it, they've got more detoxification enzymes, so that they're more feisty, if you like. So it gives us an idea of specialist versus generalist. Hopefully, so this is LC50 data coming from a whole load of bioassays that have been done. So I hope it's quite clear that you can see that PAF, the P-albumin itself, has some insecticidal activity, as does GNA towards both aphid species. But when you fuse the PAF to GNA, you get a vastly reduced LC50. Okay, to try and understand what's going on here, some microscopy. So these are aphid guts that have been dissected from aphids that have been fed, pea aphids, that have been fed on FITC labeled proteins. The red dye here is just propylium iodide. It's just a dye to show you where the gut is. So you feed the aphids for 24 or 48 hours, I can't remember exactly how long, and then you chase feed with controlled diet with nothing in it. So you're looking at how fluorescence remains within the gut or not. So I hope you can see here, we've got GNA at the top here, so this is 24-hour chase. You can still see some fluorescence in the gut. Not very visible after 48 hours. P-albumin, it's gone. The key thing here is that the fusion protein, you've got fluorescence that remains within the aphid gut 48 hours after chase feeding. So what this tells us, and the graph on the right here is just it's a graphical representation of the fluorescence. So hopefully you can see here, what we think is going on is that the enhanced oral activity is due to GNA anchoring the P. albumin protein to the gut epithelia. Okay, so contact activity, again, we're a laboratory setup, so we just place the aphid in contact with a very small volume of protein solution, and then we put them on artificial diet and look at, essentially look at mortality. So this is a whole load of treatments, maybe a little bit overcomplicated. We use a sticker, we use Breakthrough to help, which is what they use in the field, to help the protein get transported. So here you see GNA has a little bit of contact activity, the P. albumin protein a little bit too, but you have to put really high doses in. The fusion protein results in a much higher level of mortality, significantly higher. And I think what I wanted to point out here is that if you compare the green circle right on the right with the next one, it's not the combination of the two actives that is responsible for the high level of mortality, it's fusion, so it's the delivery system that's working here. It turns out, I said earlier, insects are chock-a-block with monocylated proteins. It turns out that one of the subunits of that rotary pump is monocylated. So we think this is how it's helping to anchor it. And remember, these pumps are in all sorts of membranes, nervous system, Malpighian tubules, and so on. Okay, so it's really nice, because pollinators are such a key issue, it's really nice we can look at effects on bumblebees. We follow OECD guidelines for this, so we can do what's called an acute oral toxicity test. In this case, we choose a protein dose that is 4 times the LC50 of the most feisty of the aphids, peach potato aphid. So we're feeding each individual bee hundreds of micrograms of protein, depending on the protein and the LC50, of course. Our positive control here is insecticide dimethoate. The bees drink a phenomenal amount of sugar solution, so each bee takes 80 microliters in about 4 hours, and then we look at mortality and phenotype. Nice to say that all of these proteins seem to be non-toxic to bees under these conditions, whereas the dimethoate bees all died within 24 hours. Not so nice. Okay, so just to summarise, hopefully, I'm sorry if I've rushed through this a little bit, we've shown that the P. albumin peptide is orally and topically toxic to aphids at high concentrations. Similarly, GNA itself has some insecticidal activity, but fusing that protein to GNA significantly enhances the efficacy of oral and contact insecticidal efficacy. And our bee assays suggest that none of these proteins are toxic to bumblebees. This is published work, if you're interested. We've got a paper, I think, a couple of years ago now. Okay, so it just remains for me to thank everyone who's actually done the work in the lab. We have a really nice project with a Brazilian Citrus Association, Fundi Citrus. We're doing some really nice work with them. And thank you very much for listening.
Thanks, Elaine. Really interesting. Are there any questions?
I'm sure everyone asked you this, but I think the snowdrop toxin is quite toxic to people.
No, it's not. Okay, so this goes back many, many years to a study that was published by a gentleman called Pushtai.
That's the one.
And they fed rats.
Aiming to discredit GM.
Yes, yeah, it was that set us back a lot of years actually. John actually was collaborating with Pushtai at the time this all came out. They fed rats with raw potatoes, some of them with containing GNA and some of them not. All of the rats were sick. The study was shown to be null and void, essentially. And there was a publication where they fed rats GNA for about 90 days. It was fine. So I would say GNA has been shown to be non-toxic to mammals. I mean, you wouldn't eat bucket loads of it. You wouldn't eat bucket loads of anything, really, would you?
Is there another quick question before you have to run for your train? Just one more here, I think.
Thank you very much. Fantastic presentation. I've worked with biopesticides myself, so I remember hand an example, please, just quite a bit I was just wondering, so in terms of next steps, I suppose, for your work, any thoughts regarding the formulation? Actually, these compounds in the field setting?
Okay, so I've worked with industry on several projects. Unfortunately, each time it's gone to right, we're going to commercialise this, the companies have merged and it's fallen off their agenda. So formulation we haven't done too much of. I would imagine we've tested several adjuvants, breakthrough, I can't remember them now, because it was a while ago. So I imagine it would be formulated in some way. It's not my area of expertise, but yeah, conventional formulation, I guess. You need to watch because it's a protein, not a chemical. I know there are new methods out there.
Yeah, no, absolutely.
Thank you.
Thank you, Elaine. I will let you get your train.